Hello, and thanks for joining us for today's webinar from Tecan. Worried about your cell-based assays? Remote monitoring with CNS offers complete peace of mind. I'm Ashley, Managing Editor for Technology Networks, and I'm here to moderate the event. I'm pleased to have Michael Feitel, my Market Manager for Tecan Austria, joining us today as your presenter. So a warm welcome to you, Michael. So, Tecan understands that there is a current trend for live kinetic assays. A key to success and to obtain consistent results from long-term experiments is the capability to monitor your experiments remotely. In this webinar, a workflow for cell-based assays is presented for a hypoxia-induced HIF factor analysis real-time cytotoxicity assay and for growth monitoring of microaerophilic organisms. Using Tecan's new CNS software, you can now check on a mobile device if your infinite plate reader is performing a measurement, and you can even keep track of the O2 and CO2 concentrations within the reader. Following the webinar, we will have a Q&A session, and we would welcome any questions that you may have. To ask a question, you should click the Ask a Question button that appears below the media player. Enter your email address and question, and just click OK. We will try to answer as many questions as possible during the time available. Any questions we don't have time to get to, we'll be sure that you are contacted offline with an answer. If you experience any technical issues, please also use this button to request support. So, I will now hand over to Michael for today's webinar.
Thank you, Ashley, for the nice introduction about today's webinar. I also welcome everyone currently online. And we're going to talk about your cell-based assays. Really, the CNS software from Tecan offers complete peace of mind. And if you're going to step into the webinar, I will give you a brief outline of the content. So why are we actually going into cell-based assays? What is the reason for doing so? In giving that, what are common problems researchers usually face when doing cell-based assays? And upon this, then, what is CNS? What is the common notification system, the new software from Tecan, which allows to remotely monitor your system and your experiment? To give more information in terms of the workflow solutions with CNS, we have three examples for you: hypoxia-induced HIF factor analysis, a real-time cytotoxicity assay, as well as we show growth monitoring of microaerophilic organisms. And at the end, I will give you a summary about the CNS and the features we offer for the workflow solutions for you, as well as some useful links and information from Tecan. So stepping in that, why are actually cell-based assays on the rise? We have a resource showing that when you look at 2009 up to 2015, it seems like a double feature where the basic issue is that an increase in the market will be in academia, biotech, as well as pharma. And we can basically have information that also in pharma, where usually biological screens are done, cell-based assays are on the rise because obviously they give more information content. Because obviously towards more biological relevance, you get smart decisions sooner. If you go from biochemical assays to cell-based assays using cell lysates, you bridge the gap for assays performed in model organisms with cell-based assays, of course. Because one issue is you want to cut down on animal experiments on one point, but if you go then further on with cell-based assays, they are more biological relevant than biochemical assays. They're providing an indicator for the in vivo situation actually in an in vitro setting that allows you to collect information on how drug candidates interact really with cells early on in the discovery process, and it helps you clearly to identify prime candidates to take into clinical trials and, of course, avoid the issue of having late-stage failures in drug development. Identifying crucial safety and efficacy data in the development process certainly can help pharma companies save a lot of dollars annually. And as mentioned, in vivo modeling early in the preclinical stage is driving the demand for cell-based assays, as shown in the previous slide. And more assays are developed using living cells, and this is really an issue we face and we have seen with customer requests how can we help people going into living cell assays, particularly the differentiator between endpoint and kinetic assays in giving really a great biological process information, high content, compared to cellular and non-cell-based assays. In doing so, we have discussed several issues with customers actually ready for go into cell-based assays and having really problems with the cell-based assays, and the most common problems we have actually laid out here, particularly in terms of a workflow. You're going from establishing a cell culture in a microplate with assay preparation, and that requires several washing steps, for instance, to change buffer solutions or to remove phenol red before reading a plate. Clearly, are you washing away your results when you do this manually? Further on, after assay preparation, you would like to protect the cells and even read from the cells during long-term life kinetic assay. If you don't have a workflow solution, this normally requires the transfer of a microplate between an incubator and a reader. Are you, of course, aware that the mechanical stresses and the hostile environmental changes that the cells experience lead to very low results and failure often in an experiment? Do you really detect every cell then in a long-term experiment with your plate reader? Normally, when we talk to customers, they tell us, well, our main problem when we read cells inside the microplate reader in a well, then we see that cells usually don't grow non-homogeneously, and that, of course, decreases the consistency of your results. And what can we do about this? And finally, if you go into long-term experiments, how can you actually check on your experiment? How do you know if the environment of your cell is still stable, if the experiment is still running? And normally that requires constant attention. And now, with the new CNS software, we have a solution for that. Coming back to the first point in terms of assay preparation, as I mentioned, you can maximize the cell number during assay preparation when you do a fully controlled cell washing procedure. I know that many people still are doing manual washing, and we have done a consistent validation from two different cell lines, as you've seen in this image, that when we compare manual washing with the hydro-speed washer from Tecan, which uses the cell protection feature in the software, which allows gentle washing of your cell, that although the wash efficiency is similar between the hydro-speed and the manual washing, you definitely have a much higher cell viability when you do an automated washing procedure with the hydro-speed. So you really can maximize your cell numbers during assay preparation with the hydro-speed washer. If we go on in the assay workflow in terms of protecting cells, how can you do this but also want to do a readout simultaneously? So Tecan offers with the gas control module an excellent solution. You can control your oxygen and your CO2 concentration simultaneously, and all this inside the reader, so you actually have like an incubator. This allows you don't have data gaps in between when you're shuffling normally your plate between an incubator and the reader, and the cells do not experience a temperature change or an environmental change in terms of oxygen and CO2 concentration. So the gas control module offers an excellent solution to really protect cells during the assay workflow. When you go on with the assay workflow and doing a readout, one issue is really the non-homogeneous cell growth, and that decreases the consistency of your results. So what we have done is we have developed this so-called optimal read mode in the Infinite Reader that allows you to really read from every cell in a microplate well. The optimal read is such that you basically split up all the flashes you have defined in the measurement run, for instance, for a fluorescence readout, in a certain pattern. So if we look, for instance, at the pattern over here versus here in the 96 well, all the flashes usually done in the middle of the well are now being distributed throughout the well, and thus you get a much better consistency and well-to-well uniformity of your results, and that this actually increases your sensitivity as well. So this is an excellent feature for reading cells inside a microplate well. And now, how do you actually know when you're doing long-term experiments if your experiment is still running and if the cellular environment is still stable? And this was now an action for us to develop the so-called CNS software, the Common Notification System, which allows you to monitor your instrument in the office. You can actually then initiate user actions in the lab by calling your peers, and you can watch the instrument status as well at home, on a web server, as well as a mobile device. This is really established for the Tecan portfolio, not only for the readers, but also for the Freedom Evo liquid handler family. If we go on with the reader discussion, then of course we know that all the infinite readers are in the field require this issue in terms of people want to know if the instrument is still running. So the CNS is compatible with all the readers in the field, from the 200 Pro, F500, M1000, and M1000 Pro, and requires the Magellan version 7.2 software. So if you have already an infinite reader and have Magellan software version 7.2, you can readily use the CNS software. What are the customer benefits with CNS? I've lined this out in bullet points in terms of really monitor your oxygen and CO2 concentration inside the reader with the GCM. You get information about the incubation time, how long the reader is actually running in terms how long is the experiment running. When you have initiated user actions inside the measurement run, such as to open the plate carrier to change plates, to adjust temperature, or even pipette the reagent manually, this information is then being notified inside the CNS software, and you can see this directly on your mobile device. When you're doing kinetic assays, obviously a cycle number, runs started, paused, or completed is a valuable information for you to decide what to do next. This information is also transferred via CNS. If you have an M1000 Pro and an integrated stacker, information about the stacker is transferred also via CNS. You get information about the measurement run and standard situations like power failure, online and offline status of the instrument as well. CNS notifications are sent to a web server as well as the iPhone and the iPad, and this is shown as an example here. So you can look at a web server as well as on an iPhone, iPad. The reason for that is simply that some pharmaceutical companies, they have restrictions in terms of having talking to the outside world via the internet, but still it's possible on the internet to do a web server issue with the CNS, so you can certainly look inside the web server what CNS is doing when you're not in the lab. A few examples of the CNS iPhone app status are shown in the next few slides. So the information is transferred in terms of what is the PC name, what is the Tecan application, if it's, for instance, the Freedom Evo or the Magellan software running, the signal status, the software running, what is the last comment you have put in, what is the expected runtime, of course the system time, and which user in the user management system actually has been logged in with the CNS. Notification examples are like if the software is running in general, if the software is connected to the reader, but it's in idle mode, and of course, if the measurement is in progress and running, as well as I mentioned, instrument errors and warnings, like if a measurement has not been finished, if you have been setting a wrong kinetic interval, or, as mentioned, if there are user interactions required, like changing plates or adjusting temperature, or manual injection. If you just want to quickly pipette something in a plate, you want to be notified when the run actually expects that from you. The main view of an iPhone CNS app then shows you really the current time, the duration of the experiment, and what I also mentioned, you basically get information about the comment, the cycle number, CO2 and O2 concentration. So this is valuable information when you do long-term assays and want to know how is my instrument doing. If you click on the details section here, you get basically in a different mode where you can see the overall status in terms of the complete experiment. So you get information about the cycle numbers, the custom action, and the proceedings. So all individual steps here are lined out consecutively. And this is a great feature you can use for a long-term experiment. We would like to find out which operating systems are most widely used, and please use this poll to let us know how we continue to improve and develop the CNS. So thank you for the polling questions and your answers, and now we're going right into the application sections with CNS, and we have chosen 3 examples to really show how beneficial CNS actually is in terms of the assay workflow. So we'll show monitoring and detecting hypoxia-induced HIF factors, a real-time cytotoxicity measurement over days, as well as the final assay is a growth monitoring of microaerophilic organisms, and all these assays benefit from the CNS. So the first application refers to the HIF factor, which is hypoxia-induced factor, transcription factor, also implicated in cancer cells where it's overexpressed. So it's a key feature to find out how HIF is expressed, and it's great when we basically take a look at an assay where we can see how the expression is induced by different environmental conditions. We basically used the gene laser reporter assay from LifeTech and their cell sensor cell line for this assay. 8 hours incubation at 37 degrees and at different oxygen concentrations, and we wanted to know how is the expression regulated in terms of hypoxic conditions. We used a NUNC edge plate in all experiments to avoid evaporation. So addition of assay substrate and signal detection was then performed. One step and one word further towards the NUNC edge plate. NUNC edge plate was actually developed to really do away with the evaporation issue what people usually have to deal with long-term assays. So this plate has a moid on each of the 4 corners, and each moid can actually take up 1.7 milliliter of water or buffer. And basically this leads to the fact that when you do long-term assays, let's say 72 hours, so 3 days or even more, usually on the edge of the plate, on the normal plate, you see an evaporation up to 10%, and with the thermonunc edge plate, this is cut down to less than 2%. So this is a great tool we have used and looked at in terms of long-term assays in a 96 plate well. If we then look at the results with the LifeTech cell sensor cell line, we also did an experiment compared to a standard binder incubator to see if the GCM of the Infinite 200 Pro reader basically yields the same result, and we can nicely show that. If you look at the results, basically between 1% and 0.2%, we can clearly see that a very low oxygen concentration of 0.2% results in a higher expression of the HIF factor. This is depicted by an increase in the relative activity of the 465-30 signal ratio. And this is a clear indication that, first of all, the induction of hypoxia inside the reader is sufficient to and absolutely comparable to a normal Binder incubator. And this allows you also to do a workflow automation when you use an injector system for the assay substrate addition. If we move on to the real-time cytotoxicity measurements, which we have done over days, we have done this together with Promega and the new Celltox green dye. This is a kinetic assay, and the great issue and feature of this dye is that it binds only to DNA from cells which have compromised membrane integrity. So in normal cells, there's no readout. Once the Celltox green dye binds, you can do actually an FI top measurement and do a readout. And the idea was to find out how do compounds actually differ in terms of pharmacokinetic. And so we used the Tecan HP D300 digital dispenser for contact-free dispensing of the test compounds, the Infinite 200 with the GCM for kinetic measurement. Actually, over 72 hours at 5% CO2. So this is a great issue when you have an assay over days to really look at the CO2 concentration with the CNS software to make sure everything is okay. You don't have to run into the lab, or even you have the opportunity then, if you see if the gas tank runs low, you call in and say, please have the gas tank changed, otherwise my experiment goes sour and I have to repeat it again. So the CNS really helps you to do that. So the HepG2 cells were used for this assay in terms of checking for the membrane integrity, and nocodazole as well as terfenadine were used as the test compounds to see the actions in terms of cell death over a time course of 72 hours. And if you then look at the results shown here, you see basically an endpoint. They look quite comparable. However, if you look at the two compounds' actions from nocodazole and terfenadine respectively, then we clearly see that different mode of actions, such nocodazole acting on the spindle inside a cell, acts much slower compared to a calcium-dependent mechanism caused by terfenadine. Both, of course, compromise the membrane integrity, but on a very different time scale. So this is a very vital information in drug development when you want to find out how fast is a drug actually acting inside a cell or on a cell per se. And so this is only achieved by using kinetic assays. And kinetic assays, in that sense, 72 hours results is in an excellent way to find out how a drug is actually working inside a cell. The last assay we want to focus on is growth monitoring of Helicobacter pylori. This is actually a very good feature in terms of looking at a multiplexed assay. So not only wanted we to show that a GFP transfected Helicobacter pylori actually is increased by low oxygen levels, but this is really coincided by an increase in the OD600 absorbance value, really showing that the growth curve is really associated with an increase in the GFP signal. So what we did was we used 10% CO2 and changed the oxygen concentration to 5% and no oxygen regulation. We let the cells grow for 22 hours at 37 degrees at continuous shaking, and as I said, did a multiplexed assay where we can really use an absorbance measurement and a GFP FI top measurement consecutively inside the Infinity reader. So this Walkaway growth monitoring now shows clearly that cells do not proliferate at all when no oxygen control is actually done, so ambient at 21%. Then cells start slowly proliferating about 24 hours after incubation and using 10%. And we definitely see a reasonable good cell growth only when using a lower 5% oxygen concentration. And this is nicely coincided by the OT600 measurement, where we can then say, okay, when you do see an increase in the FI signal, that's not only related to an increased expression, but also definitely related to an increase in cell growth, reflected by an increase in the OT600 measurement. So in summary, what CNS can really do for you is really boost your productivity in your lab. As a summary, you can really monitor your oxygen and CO2 concentration to check the status of the gas tank actually remotely by the CNS software. This leads to the fact that you can initiate a user action inside the lab, really call your peer and say, hey, please, I'm at home, I have no time, but I see I have a problem in my lab. Make clear that my instrument continues to run, change the gas tank. So this leads finally to the fact that CNS increases the walkaway time for you and gives you greater productivity and improves process security by the fact that with CNS, the steps notified from the CNS can be tracked, and you have the capability of following up all the steps which have been programmed inside the measurement run. So to conclude, what we see is really a necessary workflow preparation and protection, as well as detection for cell-based assays. And if you're worried about obtaining consistent results, so Tecan really offers a complete portfolio, including the hydro-speed washer for really maximizing the number of cells during cell preparation with washing, with the Infinite 200 Pro plate reader and the GCM. You have a perfect combination to maintain a perfect environment for long-term experiments, and the remote monitoring with CNS clearly helps you to ensure the high quality of your assay results the first time by looking at the O2 and CO2 concentrations, by looking at the cycle numbers, if the instrument is running, if there's a potential power failure, etc. So this gives you really an excellent tool to follow up on your assay, and as a complete solution gives you confidence that you get the right results the first time on your assay. We invite you actually to take a look at the Tecan journal, which publishes very nice scientific results from our customers. So if you have an exciting story to tell us, please let us know. Get in contact with us, and we will make sure that your assays and your results and your story will be published, and so that everyone can hear from you what you have done using the exciting solutions from Tecan. And so if you have any issue you want to discuss with us, please let us know and contact us. And finally, our partners in cell biology really help us together in terms of providing solutions to our customers for cell-based assays. We clearly see this further on the rise to really increase the information contact, to get more information with cell-based assays, more comparable to the in vivo situations. So these partners in cell biology clearly help us both in terms of 2D as well as 3D cell culture assays. And finally, I would like to point you to several links from Tecan where you find valuable information. So Tecan in general, of course, Tecan detection, the Infinite 200 Pro, and more. What I think is really interesting, I hope you will continue the journey together. Take a look at the tecan.com/reader family link. All the webinars from Tecan in 2014 will be there, and the theme about cell-based assays, also with partners and customers, will continue. And if you want to get more information and want to really register for these webinars, go ahead, and we look forward to talk with you about your essay as well if you have a particular question about this. The Tecan landing pages, of course, are referring more to the product information, particularly for the GCM and the hydrospeed, particularly the cell protection mode for the assay preparation are here discussed, and we'll give you also information about application notes where all these issues with the GCM and the hydrospeed are, of course, scientifically published and discussed in more detail. So we look forward to discuss with you your essay in terms of assay preparation, protection, and detection with the CNS monitoring, of course. We provide you a great solution to really monitor your experiment remotely. And certainly, thank you very much for your attention in this regard. And we are now ready to take your question. Thank you.
Thank you very much for your time, Michael, and I'm sure you'll all agree that was a really interesting presentation that raised some excellent points. So what I'd like to do first off is just jump to the results from the poll, asking obviously which operating system you use. It's quite an interesting results we've got here. We see 50% of respondents have replied that they use Apple iOS. That's followed by 35.7% of people using Android, and then at 7.1% we have both Windows Mobile and other. So from that we can see heavily dominated really by Apple and Android, which I suppose is no surprise really. So after covering the poll, what I'd like to do is just move on to some questions we've had in. So I'll jump onto those now. Hi Michael, the first question we've had is, do I have to pay for the CNS software?
Mm-hmm. Well, certainly an interesting question. No, basically the CNS software is actually part of the Magellan software, so users only have to do an upgrade if they're already using a Magellan version, so they need to have version 7.2, and the CNS, of course, comes then for free as an excellent add-on to the software to help users really remotely monitoring the reader.
Okay, thank you. The next question is, what is the distance range of monitoring remotely in real time for the cytotoxicity data?
Okay, interesting question. So basically that just really depends on the bandwidth on your connection. So if you look at sort of cytotoxicity data or kinetic readings, you're doing the readings every 30 seconds maybe, or even 1 minute or so. And of course, sending an email goes more or less in an instant. So of course here there's no issue in terms of losing data or losing information, because the internet, of course, is much faster than the usual kinetic assay of a reader, so there's no problem in terms of remotely monitoring your reader, depending on the distance.
Okay, sure. The next question is, for reading cells in a microplate well, I can use well scanning, so they're wondering why they should use optimal read.
Yeah. Well scanning certainly is a feature where you have the capability of looking at a very specific content in one single well, but that means, of course, that you have to use a lot of time reading in a one single well. And so the idea from Tecan was really to get optimal results in terms of well-to-well uniformity and increasing sensitivity in cell-based assays to split up all the flashes, what you usually use, and this is what we call optimal read. So that means you basically don't lose any time with a standard readout. The same time will be spent when you use optimal read, but you get more consistent data compared to well scanning.
Okay, excellent, thank you. And we have one final question. Is does the CNS software allow to control the reader?
This issue was actually discussed quite extensively here at Tecan and also with customers, and the solution was then that it's a security issue that we have decided not to allow to really control the reader remotely, because obviously an iPhone or external device can be used more or less by anybody, and so it was really a security point that we say, okay, remote monitoring gives you the capability of doing actions, really calling into the lab and asking your peers to basically change the gas tank or change the plate or whatever. But mainly the answer is it's a for security reasons we are not allowing to really control the reader remotely.
No problems. That obviously makes quite a lot of sense.
Yeah.
So thank you for clarifying that. So if there are no more questions, I'd like to thank Michael for his time, and like to thank everyone for attending. We will be putting this webinar together as an on-demand event, and obviously sharing that link. We would ask if you think of anyone in your institution or in your business that may be of interest, please feel free to share that link, and thank you very much for your time.